So you may be wondering what's going on. Hi, my name is Miracle Moses. I am a medical student trying to survive university life one exam at a time. Right now, exams are getting really close, and honestly, the pressure is real. So I thought, why not turn this into something productive for both of us? Today, we are going to do a simple 30 minutes study section together. No stress, no overthinking, just 30 minutes of focused reading by 5:30 a.m. Whether you are also in school, preparing for exams, or just trying to build a better reading habit, this is your sign to pause everything. Grab your book and join me. You don't have to be perfect. You don't need motivation. You just need to start. So get comfortable, put your phones on silent, or put them by your side while we read together. Take a deep breath and let's read together for the next 30 minutes. We've got this. Pray. All right, let's begin.
Using reliable primer and split into 4 test tubes, each having one test tube of DDNTP, liquid post-chain integration. When the form temperature is completed, the resulting mixture of intercourse with electrolysis on a polyacid gel and is dissipated according to the weight net. So that's just the method. See, that method is used for the principle of maths. It's used as a principle for DNA sequencing. And then we have the steps of polymerase chain reactions. Polymerase chain reaction. We have formula at the end of the day. They have a step. They have gradient, they have brain. Next step gradient real-time reverse transcriptase multiplex AFP real-time. Okay, so next step reverse transcriptase PCR real-time gradient multiplex and PCR. So now then the next one is steps. The steps is denaturization, right? Steps is denaturization. Now hybridization, elongation. Is the steps hybridization, hydration, and dehydration, hybridization, and elongation. And it is not dehydration. It is denaturization. Denaturization, hybridization, elongation. Those are the 3 required steps for PCR. Yeah, that's the polymerase chain reaction. And then for the application, the one is in gene, that's genetic or paternity testing. You can find it in metagenesis. You can find it in mutation, so like detection of mutation. You can find it in DNA amplification, DNA amplification. Now, what is PCR? PCR is a biochemical technology used to amplify single few copies of a piece of DNA. It is used to amplify what few or a piece of what DNA across several orders of magnitude generated down. Across several orders of generated sequence. And the principle, the double-stranded DNA of interest is denatured to what separates into 2 individual and each strand is allowed to what hybridize with a primer. The primer template is used. The primer template is used for what this DNA synthesis, and it's called what DNA polymerase and denature, denaturization, renaturization, and synthesis are repeated over and over to generate a multiple target DNA. Now, if you explain steps include there are steps that but they are required for success. And we have the primer, we have the stack polymerase, we have DNA material, and then the deoxoribone nucleotide triphosphate, DTMPs. Those are the steps and they are required. Now, the normal steps we mentioned in the beginning is that we have denaturization, hybridization, and elongation. Denaturization is the separation of 2 strands. That is, the first point you should take note is that the separation of what? 2 strands. And then there's a separation of 2 strands of DNA by raising temperature to about 94 degrees Celsius, called denaturization temperature. At this temperature, the hydrogen bonds are broken from what? Single-stranded what? DNA. Now, hybridization also carries out in a temperature, but the temperature here is about 30 to 40 degrees. It's about what? 30 to 40 degrees. And it's called primer hybridization temperature. Primer hybridization temperature. Now, this falling temperature allows the hydrogen bonds to reform. And as they reform, causing the complementary strands to what? Anion at 54 degrees Celsius. Now, elongation, this occurs at 72%. This occurs at 72%, and this occurs in that polymerase would binds to the prime single-stranded DNAs and catalyzes replication using DNTPs present in the reaction mixture. Now, in the second cycle is that 4 molecules are cutting, and it takes about 20 to 40 cycles to synthesize and what analytical amount of DNA. PCR is extremely rapid and lasts for a few hours, 2 to 3 hours per hour. 2 to 3 hours per 30 seconds. That's the reaction that you're going to expect from that. Now, explain the mode of action of substitution enzyme. The mode of action of substitution enzymes is the substitution enzyme are sensitive to methylation. The first point I should take note is that they are sensitive to methylation, and this methylation of bases between recognition bases inhibits the action of the restriction enzyme. Now, some RNA, that's some restriction enzymes, recognize tetrameric sequence, which is like 5' G80 such as such as SAL 381. So they are trying to tell that the example of this is and it's from a bacterium Streptococcus aureus, and then we have the numeric sequence such as what? Now many RE, like many of them are staggered cut into DNA strands, leaving single-stranded overhangs, or sticky ends such as EcoRI1. EcoRI1 is an example, such as when they give examples of tetrametric sequences, they give some 3 in 1. And now one is some generate what? Some generate flushed ends such as what? HPV, and some are specific and clear at any organization site. Now there are certain REs that are capable of cutting at the same site in a revolution sequence and are called isochirosomas, while other cuts at different sites are called heteroschemas. So there are different types of cuts at their particular expectation areas. Now let's say briefly explain the importance of precision enzyme. Precision enzyme has one thing that I always realize that precision enzymes are optimal in the generation of viral diseases in the context of elephant virus in the context of virus DNA stuff. And now that precision enzyme, otherwise called precision nucleases, and nucleases that it's 6:15. Let's just continue. I don't know how many minutes, but let's just be clear. Now, the first part is the precision enzyme, other called precision endonucleases, and nucleases that leave single or double-stranded of DNA sequence at specific sites known as revolution sites. So, like which I was doing, I go back and somebody called this of endonucleases, precision endonucleases, and they are found in specific sites. They prevent invasion of foreign DNAs. Such as viral DNAs by cutting it up, thus restrict the host range of what virus. And because they cut the foreign DNAs at specific sites within the DNA and not at each site, they are called what? Endonucleases. Now, precision enzymes are used in the creation of what? DNA fragments, which find usefulness in recombinant DNA technology and what? Cloning. No matter how it wants to be very, very funny and you will be, just remember recombinant DNA was biotechnology. That's where you find this recombinant DNA technology that I've seen so many places and also in cloning. The next point is the right short note on the application of biotechnology in medicine. And there's pharmacological, there's biogenomics, there's gene therapy, there's killing of specific cells. It's not here. There's genetic testing, there's genetic engineering, right? So we have the biotechnology is the technology of living things that utilizes biological systems, living organisms or part of it to develop or create what products. Now, example is biopharmaceuticals. Pronunciation. Now, the biopharmaceuticals are formed from cultured microorganisms with no chemical involved. Now, one method is used of what a bioreactor reactor, which is a container used to grow microorganisms under specific temperature and other conditions. It's just like this what do you call this stuff, like a stuff that will help you to grow plants, and they call that stuff pharmaceuticals. Now, why they call it? Because that's how they even get drugs like in penicillin. And the gene therapy, this has been used to treat and diagnose diseases like cancer and Parkinson diseases. The mechanism of this technique is the genes are targeting the antigens have to be targeted, which can either destroy and damage cells or replace them. Pharmaceutical genomics, this uses biotechnology tools to analyze the body's response to certain drugs. It uses particular things to analyze certain drugs, and it is a combination of pharmaceuticals and biotechnologies. The aim is to develop drugs which are in certain question. The method of genetic testing is to use a DNA probe, which the sequence is similar to the mutated sequence. Then we have genetic engineered isomers. And this is the isolation of humanism using recombinant DNA technology, which was also used for restriction enzyme, evidence of restriction enzyme. So it's going to separate it, and as it's separating it, the main point is that the news came when it's coming from animal sources, had allergics and what complication. So I think they wanted to use animal insulin to provide for human use, but they figured that it's not like really active, so they had to like change the choice. And then we have molecular diagnosis. The molecular diagnosis, we have PCR. PCR is using PCR techniques application of the nucleic acids in pathogens, allows us to detect pathogens at very low concentration, example detection of HIV and suspected pathogens. So they're using that PCR rate to be able to detect HIV, no matter how small or no matter how sensitive it is in terms of concentration. PCR is used, and it's under molecular diagnosis. And when you call it diagnosis, small diagnosis, and how small it is, you can be able to see it. Then you have ELISA. ELISA is based on antibody reactions. When you think of ELISA, the first question that you have antibody reaction, and this ELISA can diagnose infection by detecting the presence of antigens in patient cells or by detecting antibodies produced against pathogens. Apply the procedures involved in the construction of genetic library. The procedures involved in the construction of genetic library. Don't forget, construction of genetic library is where you see flammable something. Take note. It is one thing to know the causes. I don't know my brains always repeating. It is one thing to know the answers, and it's another thing to know the question. And it's very important to know the question so that you can be able to answer the answer. Do you understand? So now, the first one is preparing of DNA. The second one is fragmentation of DNA. The other one is vector preparation. The fourth one is ligation and introduction into the host. And the last one is storage of genomic codes. These are the things that you are meant to focus on. These are things that are meant to put up. Okay, this is what I need to do. For easy remembrance, we have pretty flamingos visible Samara. Don't forget these are the important things you should know. So the first one is what? Preparation of DNA. Flamingo, fragmentation of DNA. V6, vector preparation. Lake, ligation and introduction to what? Into the host. And the last one is Samara. The Samara is for what? Storage of the genetic DNA, storage of genetic library. Those are the procedures that are involved in genomic library. Now say list the application of genomic library. The application, that means what is the value of application of the genomic library? And we have that it helps in identification of what? Novel pharmaceutical important genes, helps in the study of genetic mutation in cancer cells. So genetic mutation in cancer cells, like genomic library helps that. It helps in the identification of novel pharmaceutical important genes. Just remember the novel pharmaceutical important genes. And then it helps in the study of functional regulatory sequences in vitro. It says as a source of what? Genomic sequence for generation of transgenic animals through genetic engineering, and it helps in determination of what complete genomic sequence of a given organism. Now, the next step that we have is outline the steps in molecular cloning. Outline the steps involved in molecular cloning. Now, we just finished the application. Now we are talking about the question of molecular cloning, and the fun thing is that you just have to know the outline. Just remember, you have to give them according to it. Molecular cloning. Remember, molecular cloning involves the choice of the host and the cloning factor. Choice of host and cloning factor. Preparation of vector, DNA, preparation of DNA to be cloned, right? Preparation of DNA to be cloned, and creation of what? Recombinant what? DNA. Creation of what? Recombinant DNA. With DNA's ligase. Choice of hosts and vector, right? Preparation of vector DNA. You have to prepare the vector DNA. You have to prepare the DNA to be cloned. You understand? So the next one is creation of component DNA with DNA ligase. creation of components with recombinant DNA like this. We have introduction of rDNA into the host organism, selection of organism containing vector sequence, screening for what code with desired DNA inside, and valid properties. So after creating the recombinant DNA with DNA like this, we should note that the introduction of what rDNA into the host organisms, you have to introduce the DNA into the host organism. And then the next one is the selection of the organism containing vector sequence. Then screening the clone with desired DNA inside and valid properties. And then the next one we have, which is the next one that we have is what describe the ways in which a recombinant DNA can be introduced into a host. They are asking you recombinant DNA. Now the topic question is recombinant DNA, right? Take recombinant DNA. How can recombinant DNA enter into the host? Don't overthink the question. Just make sure that you know that the only way recombinant DNA can enter into a host is by transfection, transduction, transformation, and electroporation. Transformation, transfection, transduction, and electroporation. Don't forget these things. This is how recombinant DNA can enter into the host. Take the recombinant DNA as heat. How can heat be passed through into a host? Please. Now, we have transformation. Transformation is a genetic extraction of cell resulting from the direct uptake and incorporation of exogenous genetic materials from its surroundings through the word cell membrane. So the transformation is just like collecting information, collecting genetic, genetic materials from the surrounding through the cell membrane. And now the next one is transfection. Transfection is process of introducing foreign bodies. Just thinking about the fact that it's transfection, you know that it involves like foreign bodies into it. That's infection of the exogenous material. And this is via viral methods that involves like stuff that are not like viral, like they're not virus, and this occurs in eukaryotic cells. Now, transduction is the process by which foreign DNA, rDNA, is introduced into another cell via what? A viral vector. This point, at this point, the first one is non-viral, the second one is viral. Just take note of the major points. Do you understand? And now the last one is electroporation. This is the use of high voltage electronic shock to introduce what? rDNA into the cell. rDNA into the cell. Now we see outline the steps leading to the production of monoclonal antibody. We have immunization, we have fusion, we have selection of hybridoma, screening of products, cloning and propagation characteristics and storage. Immunization, cell fusion, selection of hybridoma, screening of products, and cloning and propagation characteristics and storage. The next question is, what are vectors? Vectors, they are DNA, they are DNAs that carries a foreign DNA into the host. They are DNA that carries a foreign DNA into a host cell, where it replicates and produces many copies of itself as well as foreign DNA. That's virus. Virus is a DNA that takes an insect into the host. Just like the insect. And then start replicating itself as well as the human life to just be replacing the foreign DNA. Okay, it will be replacing itself as well as foreign DNA that follow to come inside. So, and there are examples of vectors. We have plasmid, we have phage mate, we have natural chromosomal DNA, so like bacterial artificial chromosome, this artificial chromosome, and cosmate plants. Then go to the last page. The last page is the characteristics of a good vector. The vector DNA should be of a good small size. It must be small in size. The vector must be self-replicating inside the host. The vector must have an glucose and restriction site. It must have a glucose and restriction site. If you have this, don't go there. You should not go there. And the vector should be easy to introduce into a host. The fact that you're easy to enter a host, you are good to go. The next one is that it must possess some markers gene so that it can be used for later identification of recombinant cells. It must possess multiple cloning sites, and an introduction of what vectors must not interfere with replication in the host, and donor DNA fragments must not interfere with must not interfere with the provision properties of infected. And then we had song with this book with Dawn. I just hope I'm not that because nothing affects me personally. I don't feel like this. So guys, I'm about to read another book. I don't know how many minutes I have remaining. You guys, I'm using the phone to film, but I know that I have to finish this soon just to end up the reading. So we're going to just take like maybe one topic and then just one topic. So what are antigens? Let's be like following from like BIC. Antigens they are substances which involve what and induces specific immune reaction in the body. And there are two types of antigens. There are antigens of two types and autoantigens or self-antigen present on the body's own cells such as A antigens and B antigens in premises. Now foreign antigens or non-self-antigens that enter into the body from outside. Non-self-antigens following what non-self-antigens receptors on the cell membrane of microbial organisms such as bacteria, viruses, and fungi, toxins from microbial organisms, materials from transported organs, or incompatible blood cells, allergies or allergic substances like amines. Now, types of non-self-antigens. Non-self-antigens are classified into two depending upon the response developed against them in the body. And we have antigens which induce development of immunity or production of antibodies, and antigens which react to specific antibodies and produce allergic reaction. So we have the one for immunity, that's immunogenicity, and we have one for allergic reaction. Those are the types of the non-self-antigens. And then for the chemical nature of antigens, we have antigens are mostly what congested proteins. They are mostly congested proteins, right? They are mostly congested proteins. And for you to know that they are mostly congested proteins, we should take note that we have lipoproteins and protein nucleotide, lipoproteins and nucleotides. That's the major thing. Then we talk about immunization. Immunization is what is the introduction. Immunization is defined as a procedure by which the body is prepared to fight against a specific disease. It is used to induce the immune resistance of the body to a specific disease. And we have types of immunization. We have passive and active immunization. Technically, we have passive and active immunization. And passive immunization, we have passive immunization is produced without challenging the immune system of the body. It is done by administration of syrups or gamma globulins from a person who is immunized, affected by the disease, to a non-immune person. Okay, that means this particular thing occurs when somebody has already had that disease and then they get the cure and then cause the immunization and then they give it to somebody that has not had the problem before. That's one thing to take note. And this is not what immune person. Passive immunization is acquired either naturally or what artificially. Now we have passive natural immunization. Passive natural immunization is acquired from the mother before and after birth. Before what? Before birth, immunization is referred from the mother to the fetus. The mother eventually gives the fetus what? Child immunization in form of maternal antibodies, mainly the IgG through was placenta. After birth, the antibodies, that's the IgG is found in the mother's womb, while the IgA is now found and are transferred through breast milk. Lymphocytes of the children or the child are not activated. In addition, the antibodies received from the mother are metabolized diet. Therefore, the passive immunity is short-lived, and the significance of this passive immunization is obtained before birth and is a prevention of what? Incompatibility in pregnancy. So we have passive natural immunization. Just remember that passive natural immunization because in mothers and child, that's what they are two, right? That's what they are two. While passive artificial immunization, this one is not developed by injecting previously prepared antibodies used from humans or animals. Antibodies are obtained from the person affected by the disease or animal form, particularly horses, which are being immunized artificially. Horses are mostly used. Can you hear me? Horses are mostly used for what? Immunization. I'll take you guys to remember. And then the syrup containing the antibodies, that's anti-syrups, is administered to people who have developed the disease. So it's always a therapeutic form. And it is what also used as a prophylactic measures. And then we have active immunization. It's acquired by activating the immune system of the body. Body develops resistance against this by producing antibodies following the exposure to antigens. Active immunization is acquired either naturally or artificially. Now with active immunization, naturally acquired above immunity involves activation of the immune system in the body to produce antibodies. It is active in both clinical and subclinical infection. So we have for the active immunization, there are natural ones call them active neuroimmunization. I think this was meant to be an active natural immunization. So you just have to activate the immunity in your system by medication or anything. Then you have the active artificial immunization. We have active artificial immunization is a type of immunization that is achieved by the administration of vaccines or vaccines. Now, clinical correlation, congenital immune deficiency disease. Congenital diseases are inherited and occurs due to the defect in B cells or T cells. So this is the clinical correlation, and we have B cells and T cells, or both. The common examples are the George syndrome due to the absence of thymus. When somebody has the George syndrome, it means that person does not have what? Thymus, which is not good. And several combined immune deficiency disease due to what? Lymphopenia and the absence of lymphoid tissues. And these are major forms in which immune antigens are produced. And another thing that's what I said the other day, that antigens are found on the what? They are found on the RBCs. So now, acquired immune deficiency, everybody knows this, which is the acquired immune deficiency occurs due to infection by some organism. The most common disease of this type is acquired immune deficiency syndrome. That is it. I feel like this is enough. I hope this video helped you. If you want more, please write that in the comments, and I'm going to be shooting more. It's going to motivate me, and it's going to motivate you. Bye. Thank you. Please like and share. Please. I beg you. Thank you. That's shame that I really will know that this is helping them. Please.